And our next speaker is Dr. Amy Proal, a distinguished microbiologist and the president and research director of the PolyBio Research Foundation in Boston, USA. Dr. Proal has dedicated her career to understanding the complex interactions between pathogens and chronic inflammatory diseases. Her innovative research, particularly through the Long COVID Research Consortium, has provided crucial insights into the persistent effects of viral infections and their role in conditions like Long COVID and ME/CFS. So please join me in welcoming Dr. Amy Proal. Hi.
Well, thank you so much for inviting me to speak. I'm Amy Proal. I'm president of PolyBio Research Foundation, and I'll talk today about pathogen persistence in Long COVID and ME/CFS, evidence and research considerations. So at PolyBio, we study post-acute infectious syndromes. And what that means is that every well-studied pathogen, from bacterial pathogens like Borrelia and Q fever and viruses like Ebola, dengue, measles, are connected to a chronic syndrome in which a subset of infected patients develop chronic symptoms that can last a lifetime. And we want to understand why this is happening and how to get people better. To do that in Long COVID, when the pandemic began, Long COVID is a prime example of a post-acute infectious syndrome. So millions of patients across the globe, as we know, are developing debilitating symptoms after the infection with the SARS-CoV-2 virus. And as much as the pandemic is terrible, it presents and continues to present a very good opportunity for research in post-acute infection syndromes, because many patients and people get sick at the same time. They get the same infection, and then we can create studies to track and study them over time to collect their samples, including tissue samples, and we can actually understand the progression of the chronic disease. To do that, at PolyBio, we formed a group called the Long COVID Research Consortium, where scientists and MDs at different academic institutions in the USA, but also in Sweden, in France, and we're sharing data, we share ideas, we meet every other 2 weeks, and we're working together to understand root cause drivers of Long COVID. These include neuroinflammation, this includes microbiome imbalances, this includes hormonal dysfunction. But early on in the pandemic, we did focus in on a major trend in this space, which is SARS-CoV-2 persistence or SARS-CoV-2 reservoir. And really, that's the straightforward possibility that at least some patients with Long COVID do not fully clear the SARS-CoV-2 virus after infection. Instead, viral RNA or protein persist in patient tissue in a reservoir, where it may continue to provoke the immune response or drive a range of other problems. And in order to best understand this trend in Long COVID, our consortium came together and wrote this paper a few months ago in Nature Immunology. And in the paper, we walk through evidence showing SARS-CoV-2 reservoirs in patients after COVID. We also delineate mechanisms by which a SARS-CoV-2 reservoir can drive different types of dysfunction and potentially symptoms. And we also delineate research priorities for the field to continue studying SARS-CoV-2 persistence, which actually form the basis of many of the projects we're working on as part of our Long COVID Research Consortium projects. Now, here's a table from our paper. These are just some of the studies that have identified SARS-CoV-2 RNA or protein in samples, often tissue samples collected from people after COVID, often with Long COVID symptoms, months or years after initial infection. Here's an example in Long COVID. This is a preprint from Tim Hendrick's group at UCSF. They got gut tissue via a biopsy procedure from patients with Long COVID, from 5 patients. And they found SARS-CoV-2 spike RNA in all 5 of the Long COVID patients' gut biopsies from 158 to 676 days following initial COVID symptom onset, with symptom primarily observed in cells located within the lamina propria. And this persistence of SARS-CoV-2 in the gut is a major trend that we're studying. The gut is dense with the ACE2 receptors that the virus uses for cell entry. SARS-CoV-2 reaches the gut in a high number of patients, so it's a strongly potential reservoir site in people. Now, as you can see, that was a tissue sample that the team had found of the RNA and protein in the RNA. And what is important is that this means that in Long COVID, and this goes for all post-acute infection syndromes, including ME/CFS and post-treatment Lyme disease, we cannot just collect blood and fluids from patients because viral protein or RNA is not always there. In fact, over time, there's a tendency for pathogens to leave the blood where the immune system is often the most robust and move, in simple terms, to hide in tissue where they're better protected. And that's what we're calling a reservoir. So it's important then to collect tissue samples from patients in addition to blood and urine. And we're doing that as part of our Long COVID Research Consortium. We have teams collecting gut tissue, lung tissue, lymph node tissue, even bone marrow biopsies from patients with long COVID. Now, there's already clues, and there's several really big papers out on the immune response and viral persistence in long COVID. This is one from Peter Broden's team at Karolinska. What was interesting is they found elevated SARS-CoV-2 specific IgG responses to the spike protein in patients with severe long COVID. And interestingly, this was inversely, the elevated SARS-CoV responses to SARS-CoV-2 IgG were inversely correlated with clonally expanded memory CD8 T cells, indicating that restrained clonal expansion enables the viral persistence. In other words, the T cells don't seem to be responding maybe potentially as correctly as they should. So this is beginning just showing you that there's a trend to understand how and why the virus persists in certain patients with long COVID. Now, to give you a better idea, just in the rest of this, it's a short presentation. There's actually so much data on persistence. I could talk for a long time. What I think would be helpful is for me to frame SARS-CoV-2 persistence within the context of other findings in the long COVID space so that people can see a big picture of how it can impact different aspects of the disease process. So here are some abnormalities that teams, including those working with our consortium, have identified in long COVID: platelet hyperactivation, increased neutrophil extracellular trap or NET formation, fibrin amyloid microclots that Ricia talked about this morning, hormonal imbalances, neuroinflammation, central nervous system inflammation, vascular problems, endothelial dysfunction, and autonomic dysfunction. All right, so let's go back to this picture, this image from Tim Hendrick's paper. This is a long COVID patient who has SARS-CoV-2 spike RNA in their gut tissue. What we think might happen is that the inflammation associated with this gut reservoir may wear down the lining of the gut, the epithelia barrier. And then potentially protein created by the virus, like spike protein, could leak into the blood, where there it can potentially be measured. If the team has tools that are sensitive enough to identify small amounts of viral protein. And indeed, several groups here, just some of these studies on this slide, have identified SARS-CoV-2 proteins in the blood of long COVID patients. There's variability among these studies. Definitely the assays used in these studies require further iteration. That's part of what we're actually working on with our program too, to best determine consistency among the different tools being used here. But overall, from 10 to up to 60% of long COVID patients have SARS-CoV-2 proteins in their blood circulating at any given time. And the thing, though, is it's not necessarily a biomarker, because this is an image taken from David Walt's team, who is using a sensitive tool called Simoa to identify SARS-CoV-2 proteins in long COVID blood. And you can see that these are long COVID patients, and they look for SARS-CoV-2 proteins in their blood at different time points. And some of the patients had protein at one time point, but not at another time point. And then they had protein at the next time point. So there's variability in how spike or other proteins seem to leak into long COVID blood. Potentially that could reflect activity of different activity of the virus in a reservoir. So for example, there might be periods, maybe stressful periods, where the virus actually creates more and translates more proteins, and other times when it's in a sense dormant and less protein is created. This is still an area of research. Overall, though, if spike protein leaks into the blood of a long COVID patient, you might get neutrophil extracellular trap formation. So what happens is that the innate immune response, one of the prime cells are neutrophils. And if neutrophils recognize a pathogen or a pathogen protein like spike, they'll swim towards it and they capture it. And when they capture it, they create these projections, and those projections are a neutrophil extracellular trap. They grab onto the protein, and now you can image these traps, as was done here by Mike Van Elzeker's lab at Harvard using microscopy. So this is blood from a 42-year-old woman with long COVID. In the left image, that blue are these nets, the neutrophil extracellular traps. On the right, in the pink, that's a stain for activated platelets. And what's really interesting here is the activated platelets are completely co-localized with the nets. Now, platelets are small cell fragments in blood that form clots and prevent bleeding. But they're also part of the immune response, and they can engulf viral and bacterial pathogens. So they've been shown capable of internalizing HIV, dengue, hepatitis, several viruses. And this team showed, this is data from Morgane Bombcell and team at the French National Research Center. This was presented at an HIV conference, CROI. This is early data from their team, so they still are, following up on this. But it's still very interesting because what they showed is they found that platelets from long COVID patients contain spike in viral RNA. So the platelets themselves may be infected in some patients with long COVID. That could potentially underlie part of their activation that was noted in the previous slide. Now, this is a study which used a digital transcriptomics approach to find different SARS-CoV-2 transcripts, RNA transcripts in the blood of long COVID patients, and several viral RNAs were upregulated: nucleocapsid, ORF7, and ORF AB RNA. Overall, the interesting part here is that the team worked on whole blood, and whole blood contains platelets. So when they found SARS-CoV-2 RNA in the blood of these long COVID patients, it's not for sure, but it could reflect that they're potentially capturing some of the RNA that could persist in platelets. So persistence in platelets is an important trend moving forward. Now, the same team that identified, this is again Morgane's Bombcell's team. I borrowed from a poster again from an HIV conference. Platelets in the body are created by large progenitor cells in the bone marrow called megakaryocytes. And they found SARS-CoV-2 spike protein in double-stranded RNA indicative of viral replication in long COVID megakaryocytes, these bone marrow-derived cells, suggesting that the bone marrow may be a reservoir for SARS-CoV-2 in patients with long COVID, especially potentially patients who are very sick. So they also found that peripheral serotonin was reduced in their long COVID cohort. And this could be due to the simple fact that platelets harbor and carry serotonin. So if they're directly infected, that's going to modulate, potentially drop serotonin levels. This team also, this is a team at Penn, Maya Levy's group, also part of our consortium. They found that also serotonin was reduced in their long COVID patients. And interestingly, a subset of patients in their study also continued to shed SARS-CoV-2 RNA, indicating viral reservoirs in their gut. Then, in a series of experiments in mice, they found that persistent viral RNA stimulated interferon signaling. And this interferon signaling led to inflammation that reduced absorption of the amino acid tryptophan in the gut. And tryptophan is a building block for neurotransmitters, including serotonin, which is another reason that these serotonin levels might be decreased in long COVID blood. So overall, a takeaway is that changes in hormone signaling can be directly connected to viral persistence, and we don't necessarily need to study those trends always separately in this field. Now, shifting gears to what's happening in the long COVID brain, this is a study published just maybe 2 weeks ago by Mike Van Elseker's group again at Harvard. And they used a PET imaging technique that can identify activated glial immune cells in the brain, which are part of neuroinflammation in a group of long COVID patients. And they found neuroinflammation across multiple brain regions, including some brain areas that are exposed to circulating blood factors via gaps in the blood-brain barrier of the brain's blood vessels. Now, this compelled them, I think this is an intelligent experiment. Before the patient got in the scanner, they collected blood and they measured factors in their blood related to vascular blood vessel health or damage. And they found that 6 of these factors, and I put 2 up here on the slide, showed a strong correlation with neuroinflammation in the brains of the long COVID study participants. They included, which is here on the slide, fibrinogen, which is a protein involved in forming blood clots, and SL-selectin, a molecule that helps immune cells attach to the vasculature or blood vessels during infection. And that just takes us back to SARS-CoV-2 proteins in blood because, for example, this is a study done by Ricia Pretorius' team. They showed that SARS-CoV-2 spike protein can induce fibrinogen or clot formation. So again, viral persistence just kind of continues to be a factor that can really underlie a lot of what we're seeing here. Now, this is another imaging study that I want to bring up. This is again Tim Hendrick's team at UCSF. What they did is they used a high-resolution PET imaging technique to identify T-cell activation throughout the bodies of long COVID patients. They used a novel radio tracer called 18F-ARG, which is a selective tracer that allows for the anatomical quantification of activated T lymphocytes, T cells. And in patients with long COVID symptoms, they observed higher tracer uptake, indicating T-cell activation in the spinal cord, the hilar lymph nodes, and the colon rectal wall, again, that gut of participants. And in some post-COVID participants, they saw this elevated tracer uptake indicative of activated T-cell inflammation in the bone marrow. So again, bone marrow is really important here. Now, what about autonomic dysfunction, flu-like symptoms, pain? we have to talk quickly about the vagus nerve. The vagus nerve is an important nerve. It innervates every major trunk organ of the body, the gut, the heart, the lungs, and then it connects to the back of the brain at an area called the dorsal brainstem. This is a figure from a paper that Mike Van Alziker and I wrote in 2021 on long COVID disease mechanisms, and it shows those purple and yellow bars are overlapping nerve bodies in the brainstem. For example, the nucleus of the solitary tract or the dorsal motor vagus. And these overlapping nerve bodies in the brainstem control what's called the Cygnus behavior response. They control pain and nausea signaling, and they control autonomic functions like heart rate, breathing, the ability to move from sitting to standing without getting dizzy. So if the brainstem signaling is thrown off, what's going to happen is you'll get flu-like symptoms, pain, nausea, autonomic symptoms. Now, the way that this works with vagus nerve is that vagus has chemoreceptors, and if it senses ongoing infection or inflammation in the body, it will convey a pro-inflammatory signal to the brainstem to stimulate that sickness behavior response that mounts flu-like and malaise symptoms, to stimulate the pain, nausea, and potentially autonomic type signaling. So if you have, long story short, if you have a SARS-CoV-2 reservoir in an area like the gut, vagus nerve can sense that, and that may affect brainstem signaling in a way that can result in sickness, pain, nausea, and autonomic symptoms. So in other words, we have mechanisms by which reservoirs in different parts of the body, as long as that body site is innervated by the vagus nerve, can impact some of the most common symptoms that long COVID patients and also ME/CFS patients develop. Now, one of the ways that this signaling of the brainstem vagus axis could be thrown off most, resulting in the flu-like autonomic nervous system pain, autonomic symptoms, is if the vagus nerve is directly infected. And that's an area of ongoing research for several of our teams in long COVID. But this is an autopsy study from acute COVID, and they did find SARS-CoV-2 RNA in the vagus nerve of every single person whose vagus tissue was examined in the study. So SARS-CoV-2 can infect the vagus nerve. The question is still just ongoing of whether vagus itself can be a site of reservoir. Now let's quickly jump to ME/CFS. ME/CFS is a condition in which patients develop debilitating chronic symptoms, flu-like symptoms, malaise, post-exertional malaise, often after infections. So for example, enterovirus infection, herpes virus infection have been connected to ME/CFS. Also stressful events often begin as an onset for the condition. And when part of how ME/CFS came to be was that there were actually outbreaks starting even in the 40s, going all the way through the 60s, the 80s, 1980, in which different groups or clusters of people developed symptoms characteristic of ME/CFS. And when, the tools that we had to go, that scientists had to examine these patients were pretty primitive, unfortunately. But several of these outbreaks, this is a paper from the Lancet published in 1956 on ME/CFS. Several of the outbreaks were tied to the enteroviruses or viruses in the enterovirus family, like Coxsackie viruses, polioviruses. And this is really interesting because the enteroviruses have many parallels to SARS-CoV-2. So like SARS-CoV-2, they're single-stranded RNA viruses. Like SARS-CoV-2, they're acquired via respiratory infection. But the virus also often gets into the gut, where it can also cause symptoms. Like SARS-CoV-2, people who get acutely infected with enteroviruses can develop a severe flu-like disease, or they can have a mild or even asymptomatic case. So one of the studies that I think is most important in ME/CFS is
quick info, 1 minute, 1 minute left.
Perfect. Okay. Is this study by John Chia's team? It's 2008, but they got gut tissue from patients with ME/CFS. And 82% of these gut biopsies stained positive for enterovirus VP1 protein within the parietal cells, with just 20% of the controls staining positive. So we're looking at enterovirus gut reservoir in some patients with ME/CFS. And that's interesting because you could see a mechanism by which persistence of enterovirus in the gut, let's say, would be also sensed by vagus nerve, conveyed to brainstem, and could also result in sickness, pain, nausea, and autonomic symptoms. So this doesn't have to be every case of ME/CFS, but this is an incredibly important parallel between long COVID and ME/CFS that many of our teams are following up on now to study. And this is a paper from Mike Van Elseker that he published in 2013. He hypothesized that the vagus nerve could be directly infected in patients with ME/CFS. This is years ago. And this paper stands as strong today as it did then. That is still a possibility. Herpes viruses, enterovirus, these viruses could infect vagus nerve in ME/CFS, which would result in many of the symptoms that patients have. So ongoing area. Last but not least, myalgic encephalomyelitis. Myalgic stands for muscle pain. Encephalo, brain, myel, spinal cord, itis, inflammation. So we have swelling of the brain and spinal cord as just the most important, symptoms historically of this condition, which means that this T-cell imaging spinal cord that Tim Hendricks' group is doing is vital to do in patients with ME/CFS. We're working to make that happen. So that's it. Here are some of the more priorities that I would say are takeaways for persistence in this field. We need to stop saying that long COVID or ME/CFS are a mystery. We have these really clear leads on disease mechanisms, including persistence. I think that's important. We need to collect and study tissue samples in addition to blood and other fluids from patients with long COVID and ME/CFS. We need to keep using these high-resolution imaging studies to understand both infectious and inflammatory activity in the brains and spinal cords of patients with long COVID and ME/CFS. We need to keep developing sensitive assays that can find small amounts of pathogen proteins and fluids that we can potentially use as biomarkers. And we need to set the stage for treatments that support and not necessarily suppress the immune response. For example, in HIV, teams are using immunotherapies that activate T cells, that activate natural killer cells to target persistent virus. We need to bring these into long COVID and ME/CFS. We are also working to do that. I think that's so important as we move forward in thinking about treatment. And thank you.
Thanks a lot, Dr. Amy Proal. Wow, so much dense and inspiring data. And we could go on, I think it could go on for quite some time, where we have, unsurprisingly, 50 questions waiting for you. So let's move there. What lab work do you recommend getting done for long COVID and ME/CFS patients to check for the pathogen resistance? So you said that it's not in the blood or it's not detectable in the blood. So is there any lab work that we can do?
So it's not necessarily not detectable in the blood. So there are teams that have found viral RNA in blood. And there are teams that have found protein in blood. The first consideration, though, is we don't know how reliable it is. So we don't know, for example, if someone were to go to measure a person with long COVID and we looked for RNA or protein in their blood and they're negative, we don't want to interpret that necessarily and say this person doesn't have a SARS-CoV-2 reservoir, because they could have a reservoir in their brain and it could just mean that the RNA and protein are just not in their blood, right? So it wouldn't be a pure biomarker of persistence. You would have to consider that it's just something you can measure to try to infer persistence. Now, the tools, though, that people are using to measure protein and RNA in long COVID are not available in the average doctor's office right now, which is a huge problem. David Walt's team that developed it's a Simoa. It's an assay that can find tiny amounts of spike protein. They're working on that, but they're just a lab at Harvard, one lab using this technique right now. So one thing that's really important and one thing that I think personally would be a huge priority for the NIH or for government agencies is to prioritize the funding for teams that can find these proteins or RNA potentially in long COVID blood or maybe in tissue samples even, and to help accelerate the translation of those tools into technologies that can be used in the clinic. Because right now, there is no way that I can have your doctor order the spike protein test. It can only be done in these specific research labs, right? So that's the problem right now, is that there really isn't something great that your doctor can order. The main thing that maybe they could order would be, IgG, IgM to spike protein in blood, which could give you a little bit of a clue. If it's elevated, maybe there's still persistence.
Yeah, so you can get a clue, but don't get a definitive.
It's a clue, yeah, exactly.
Yeah. So there might be a virus in our bone marrow. The question is, can we get it out? Can we use antivirals there, like in HIV? How do we deal with it?
Yeah, that's a huge priority for our consortium right now, is thinking through how we're really going to treat SARS-CoV-2 persistence. So there are some clinical trials happening now for trying to target SARS-CoV-2 reservoirs in long COVID. There are 4 teams trialing Paxlovid, which is great. There's a monoclonal antibody study at UCSF, which is cool. They're also trialing another drug, antiviral Incitrivir, which is great. The only thing that we know, though, is that a monotherapy and a shorter course of an antiviral, for example, like Paxlovid, might not be enough to clear a reservoir, because in HIV, for example, or just treating other infections, usually a combination of drugs is really needed to get at the infection. In other words, you have to come at it from multiple angles. And one of the things is that antivirals often prevent virus from infecting new cells, or they block parts of viral replication, but they don't totally get rid of the virus. So what helps is if you have another therapeutic that can come in and support the immune response to come in and kind of clear that persistent viral RNA. Monoclonal antibodies are potentially something that can be used in that capacity, but I mentioned those immunotherapies, the natural killer cell or T cell activating immunotherapies. Again, those are going to be key in this space. We're working on starting the first immunotherapy trial at UCSF in long COVID. We actually hope to announce that in the next few days. And I think that that's going to be key in trying to get reservoirs out of really difficult areas like bone marrow, if that's the case.
Okay, so exciting news to come, hopefully.
Yeah.
You mentioned lots of angles of attack to addressing the persistent virus. And one question is, someone needs to tell the NIH. So are you in contact with them? And why are we still spending millions on exercise and meditation? Bit of a provocative question there.
Yeah, I mean, I think that one of the things that is necessary for, long COVID is an emergency, as is ME/CFS. I mean, there are millions of patients. People are bedridden. Their lives are, complete. They're losing family, jobs, everything. The effect on the economy is incredible. The effect on the workforce. Under those conditions, we need to just fast track what we think is most likely to be happening. In other words, that's why our consortium focused highly on SARS-CoV-2 persistence. We're studying all kinds of other things in long COVID. If you look at our website, we're looking at all kinds of, there's microbiome studies, there's, studies on neuropathy. But overall, when we saw evidence for SARS-CoV-2 persistence, we went in that direction. And I think what NIH needs is they need to have more vision and leadership so that they have a person in a role at NIH who really just sits there looking at the big picture of the top trends in the space and then makes sure that what moves forward really are those top findings and the consequences of those. So I think that NIH needs, a visionary person that can help direct a bit more of what they're doing.
Yeah. Well, it's a big task, I guess. Yeah, but worth pursuing. Thanks a lot. Please elaborate on the connection of viral persistence and mast cell overactivity seen in long COVID, as not all long COVID will have traces of the viral persistence, or we cannot measure them. But what would be your proposal to explain their symptoms then?
Yeah, mast cell activation would just be completely connected to persistence. And in fact, mast cell activation is connected to persistence of many pathogens. If you don't clear a pathogen, it's likely that the innate immune system especially continues to recognize it to a certain capacity. And mast cells are like pawns in a chess game. So if you have your different, chess game pieces, pawns just go out there and they try to respond to everything. That's mast cells, right? So if you have an unaddressed infection, mast cells are going to be there and saying like, hey, this is still here. Hey, this is still here. Hey, this is still here. And they're just going to be degranulating. So if, and then they become primed. Now they're in a state where they're already, they're active in the next thing that stimulates mast cells. It doesn't even have to be an infection. It could be something else. It could be a food. It could be anything. Now the mast cell, because it's primed potentially, because it's responding still to the persistent infection, is going to respond even more dramatically, potentially to the next thing. So yes, there's hope that getting rid of reservoirs could help, mitigate mast cell activation. Conversely, though, as we think of treatments, certainly calming down mast cells with mast cell drugs to just try to better function makes sense, right? But at the end of the day, yeah, mast cell activation can definitely be connected to persistence. So we shouldn't study them totally separately. It's one of these things, like, what I think really matters in the long COVID and ME/CFS field is we need to unify topics when we study them. Right now, often there's a lab that studies persistence and there's a lab that studies mast cells. Well, that's good, but we got to bring those two labs together to start to understand the intersection of those things. And that's important because there is.
Yeah, thanks a lot. So the mast cell as the canary in the coal mine that can actually detect pathogens that might be there. One last question, and we need a very short answer for that. Do you see ultimate elimination of the viral reservoirs in the future? So what's your prognosis here? And please give us some hope.
Yeah, look, the top teams in the world are working on this, and they're so motivated to get stuff done. We have a huge amount of experience in HIV and cancer to pull from in treating persistent infection. There's drugs out there. We have, in other words, the talent and the resources. It's just a matter of funding and being able to move forward. But I do think that with enough support, we can move forward clinical trials and a good amount of research that will help us really understand what's going on. Yeah, so
thanks a lot.
I'm optimistic that, it's yes, we need resources. Like right now, we do have private funding at PolyBio for our consortium, and that's great. That's helping us move forward. But it would be helpful if there were also government funding opportunities as well, just so that if there's a combination clinical trial or something for reservoir that we can't always swing with private money, we could also feed it into some of those things too, right? So again, that's important to communicate. I'll just say it one more time that we know what to trial, right? We just need the support to do it.
That's a good statement to close this, but way too soon we reach the end of our allotted time. Thank you so much for sharing your expertise with us today, and looking forward to your information in the next few days that you teasered for us. Thanks a lot.
Great.
Have a great day.
Thank you.